Good evening, everyone. I, Ritika Jaiswal, on behalf of Concord Biotech, it's my extreme pleasure to extend a warm welcome to all the participants who have joined for this scientific session. Concord Biotech, the global biotechnology and immunosuppressant expert with strong capabilities in manufacturing of high quality APIs and finished formulations. With a presence in more than 70 plus countries, we are committed in delivering quality products in therapeutic segments like rheumatology, immunology, transplant, and nephrology, driven by a patient centric approach. With this commitment, we welcome all of you for joining this scientific session. And once again, it's our extreme pleasure and honor to have our eminent speaker, Dr. Vish Prakash Tiwari, sir. A very warm welcome to you, sir.
Thank you so much. Thank you so much.
Dr. Vish Prakash, as we all know, he is a renowned rheumatologist based at Indresh Hospital, Dehradun, and he is continuously initiating uh, a continuous series of rheumatology sessions and immunological disorders. And today is our privilege that today is the seventh session of this series. So, on behalf of all the participants, first of all, we thanks Dr. Tiwari and let us take this session forward. Thank you, sir. You can start the
Thank you.
session, sir.
Thank you very much. Okay, today we will discuss the T cell in rheumatology. So, first of all, we have to understand basic concepts like what is T cell? That is one. Then how T cell develop? That is second. Then how T cell becomes activated? That is third. Then fourth one is how memory developed and how different types of the T cell developed. That is fourth. Then this signaling and everything, signaling and how this integrate with the diseases. So let's see what is the T cell. So this is a lymphocyte, such as B lymphocyte. We have a T lymphocyte. It's a immune coordinator. It's a basically immune coordinator. It coordinates with different cells like B cell, macrophage, T regulator. We will see the coordination with the cells. Second, it has a receptor. So this receptor, there is a recognition of the receptor with antigen presenting cells. And downstream, there is lots of molecules which involve in the signaling or activation of a T cell. So there is a antigen recognition, that is first part. Then signal integration, that is second part. Then differentiation and communication, that is third part. Fourth is tissue impact outcome. So tissue. And T cells are not functionally, T cell is not purely phenotypically divided. This is actually a dynamic phenomena. So, although we have different types of the T cells, we will see कि it has dynamic phenotype. It has a phenotype, but it's a snapshot. Basically, it's showing a dynamic phenomena. Although each T cell can behave like other T cell, other variety. So this is T cell. We will discuss in the subsequent slide. Uh, so how the T cell develop? In bone marrow, T cell develop. That's good. But T cell got education or the development is occur in the primary lymphoid organ that is known as thymus. So this is thymus. Thymus is a capsulated structure situated in the mediastinum, in the anterior mediastinum. Up to the age of 12 or 13, it got involved. So this thymic structure has a cortex, has a capsule, then cortex, then medulla. This is thymus. How this T cell initially developed in a bone marrow, then goes to the thymus. How this goes to the thymus? Because there is a coordinated signal. So T cell has a receptor of CXCR4 and thymic stromal cell, they are secreting some ligand that is CCL25. Just one thing, there is one ligand, one receptor. Because of the gradient, T cell is going towards the thymus. So initially T cell form in the bone marrow, then it goes to the thymus that is there. So what is the roadmap of a T cell? So once T cell reach to the thymus, this is a purely precursor T cell. Then precursor T cell goes under the first stage that is called as double negative stage because we know T cell broadly we think it's a CD4 or CD8. That is not true. But initially it is double negative stage, means both CD4 and CD8 is negative. Then there is a beta selection that is for the receptor. So beta selection occur. Once the beta selection occur, proper receptor form, then they forms the double positive state. That is two or both CD4 and CD8. Then T cell rearrangement, then single positive state, then outside. So this is the developmental route. So what occur in the cortex? You have to understand from this slide only two things: positive selection and negative selection. Initially, T cell, once it is in the cortex, they got education from the cortical thymic epithelium cell, and they presented the self antigen. If the this primitive T cell able to recognize this antigen, then this will survive. This is called positive T cell selection. If they doesn't able to recognize this, they can go under apoptosis or energy. So this is the first stage in which we are educating the T cell at primary level, where whether the T cell can a T cell can recognize T cell can recognize the self antigen, that is called positive selection. Then the T cell migrate into the cortex through the again cytokine gradients. A T cell is expressing CCR7 and then ligand is 19 and 21. Then they goes to the cortex. In cortex, before going to the cortex, there is a lineage commitment. What is a lineage commitment? This double negative T cell becomes the double positive T cell. Then from the double positive T cells, only those selected who is either acting towards the MHC1, restricted towards the MHC1 or MHC2. So after the restriction, this is called lineage commitment. T cell goes into the medulla where we have a dendritic cells and other thymic medullary thymic epithelial cells. Then they again show the self antigen, and those T cells which are very aggressively attached to the self antigen, they got deleted. Those who got attachment, the sustained attachment, they can convert into the T regulator cells. Or those who doesn't attach, they got selected for the immigration. So in the cortex, you can see positive selection, that is which able to which T cells which is capable enough to recognize the self antigen at least without killing it, without activation. Then there is a lineage commitment, either CD4 or CD8. Then again, there is a double check, again showing a self antigen. If they are reactive to the self antigen, that is called negative selection. The positive selection is for the those T cells which you are selected for the further development, and negative selection for the those T cells which are self-regulatory, self-reactive. So outcome is either one is deletion. How we got rid of these autoreactive T cells? Through apoptosis or regulation, because I told you, because of the T regulator cell is also there, they can also regulate this. So this is this part for the T cell regulation. Then the roadmap is simple. If suppose there is no positive selection, the death is by neglect. Then if there is very optimal signation, then there is a positive selection, either CD4 or CD8. Then strong self recognition leads to the deletion. So what I told, what I want to convey, in the thymus, there is a first tolerance or first checkpoint. So body wants to prevent this autoimmune T cells, but this is not perfect. That's why we have peripheral T regulation also. So this is development in the thymus. Main part is this diamond development occur from the cortex to towards the medulla. Cell goes from the double negative stage to the double positive stage to the single positive stage to the perfect T cells. T cell can be undergoes through the positive selection and goes under the negative selection. And final word is that this autoimmunity regulation, this checking up, this tolerance is not perfect. Then how this T cell recognizes the antigen. CD8 recognizes the antigen through MHC class 1, and CD4 recognizes the antigen through MHC class 2. So MHC class 1 always important for the intracellular pathogen. Intracellular pathogen means who has already all developed intracellularly or they are intracellular like a virus. So this is through MHC1. So once the cell antigen is intracellular without phagocytosis, directly intracellular, then through the proteasome it got degraded. And through the TAP protein, that is a transporter activator protein, it goes into the endoplasmic reticulum. And in the endoplasmic reticulum, it attaches to the MHC1 and goes to the surface. So MHC1 presents the intracellular antigen. MHC2, if any extracellular antigen, anything coming from the outside, so this got phagocytosis goes inside. So antigen uptake by the phagocytosis, then processing in the lysosome. In lysosome, this antigen got chopped. Then they loaded on the MHC2 and they presented to the T cells. So main point is this: MHC1 is for the CD8 cell, MHC2 is for the CD4 cell. One exception is their cross presentation. What happens? Suppose extracellular antigen is presented by MHC1. That is called cross presentation. So in cross presentation, uh, this CD8 cell get activated from extracellular antigen also. So this is their MHC1, CD4 for the MHC2, CD8 for MHC1. Then there is a cross presentation. Then module 4, this signal. So three signals are there. First signal is for recognition. Recognition through MHC molecule. So this is MHC molecule, this is antigen, this is T cell, and this is peptide. So MHC molecule, peptide, T cell. So correspondent T cell attached to this. This is called recognition. And second is for co-stimulation. Once this got recognition that is not sufficient for the activation of this T cell. So it got the permission. This is called recognition. This is called permission. In permission from the antigen presenting cell, co-stimulatory molecule like CD80, CD86, they interact with the CD28, which present on the T cell. That got the permission. So T cell now going to be active. What is the fate of a T cell? What type of phenotype of a T cells it will convert? It will depends upon the cytokines. So cytokines released from the antigen presenting cells or from the all from the surrounding nuclei. It can convert change to the T cell phenotype that is dynamic. So first is recognition, second is permission, third is differentiation. So we will see that T cell fed. T cell can be Th1, Th2, Th9, Th17, Th22, TFH, T follicular helper, T regulator. But these T cells are not only these T cells. There are other T cells like memory T cells, regulatory T cells, tissue resident T cells. These are the important. So and this, if co-stimulatory molecule is like CTLA or PD-1, this is called co-inhibitory molecule. So suppose after the recognition, if permission is not there, then this T cell will go under energy. Then will not become active. So this is another mechanism of a peripheral tolerance. So three signals are important: recognition, permission, and differentiation. Then how once the permission got means MHC attached to the TCR receptor, then co-stimulatory molecule attached to the CD28. Then the downstream cascade starts. So this downstream cascade, once this antigen is attached, so these downstream cascade, they form the signalosome. Signalosome means one intracellular protein that is made up of JAP70 and LAT and SLF76 means what is there? Once the T cell got activated, the downstream pathway start from the CD3 part, which has a ITAM molecule. ITAM means tissue immune activator. So they form the phospholipase gamma C and phospholipase C. So phospholipase C breaks the membranes inositol triphosphate into inositol diphosphate into the inositol triphosphate that attach to the receptor on the endoplasmic reticulum that leads to the release of calcium channel that leads to the activation of calcium calmodulin pathway that is called NFAT. So once the T cell got activated, one molecule is NFAT. Second, phospholipase C breaks the inositol diphosphate into inositol triphosphate and diacyl glycerol phosphate. These DAG activate the protein kinase C, which activate the nuclear factor kappa beta. So NFAT is for the IL-2 and other co-stimulatory molecule. Nuclear factor kappa beta for the inflammatory cytokines like IL-1, IL-6, TNF alpha, co-stimulatory molecules, and other. And AP1 is a growth factor that is for the survival. So once they got the signal to permission, they becomes activated. So they want proliferation. Through which they will get the proliferation through IL-2. So they are getting IL-2 through NFAT. They have to create the inflammation. That inflammation will be created by nuclear factor kappa beta. Then there is a cell sustenance that is got by the AP1. So this is a channel, and in this pathway you have to remember JAP 70, because in many rheumatological diseases you will map, oh, this overactivation of JAP 70 can lead to the excessive NFAT nuclear factor kappa beta and AP1, which can lead to the hyperinflammation or autoinflammation. Then once this cytokine signal 3 now come to the this part, comes to the differentiation part. So once cytokines bind to the this is a such as go from the signal 1 recognition, signal 2 that is permission, co-stimulation, signal 3 is a differentiation. So this antigen after antigen co-stimulation cytokine, this cytokine leads to the signaling through NFAT, AP1 and nuclear factor kappa beta. And they create the transcription factor, and transcription factor leads to the different types of T cell. Suppose cytokine combination is IL-2, IL-12. Then that transcription factor will be T-bet, and this is a stat for molecule leads to the TH1. Then this is, suppose IL-4 is there, then transcription factor is GATA3. T-bet, TH1, GATA3, TS2, cytokine IL-12, here IL-4. TH17, TGF-beta, IL-1, IL-6. ROR gamma T is a transcription factor. Then T follicular helper, IL-6, IL-21. Transcription factor BCL6. T regulator, FOXP3. T9 PU1. So you have to just remember this AHR. AHR means aryl hydrocarbon transcription factor. That is a master transcription factor for TH22. That is important for the ankylosing spondylitis part. So, and another CD4 cell, CD4 CTL cytotoxic cell, the EMOS is there, BLIP is there, that is for IgG4. So just remember the transcription factor. T-bet is for TH1, GATA3 is for TH2, ROR gamma T for TH17, BCL6 for TFH, FOXP3 T regulator, PU1 TH9, AHR TH22. All these are the transcription factor. Now, how the trafficking occur, because T cell is traveling into the vascular lumen, and T cell has to reach to the inflamed tissue. So initially there is a adhesion rolling. The rolling is through selectin. After the selectin, there is a some chemokines that leads to the integrin stimulation, and then there is a arrest, integration activation, then fermentation, then diapedesis. So this is there. Just remember selectin, cytokine, integrin, diapedesis. So then diapedesis is what? This is a chemotaxis, directional flow. Directional flow, how they are occurring? This is through the receptor and ligand. So just remember CCL19 and 21. These ligand are secreted by the inflamed tissue, binds to the CCR7 that is present on the T cell. This is important for the T cell homing in the lymph node. So for the lymph node, you remember CCL19 and 21 ligand from the lymph node binding to the CCR7 receptor on the T cell. That is important for calling T cell to the lymph node or primary lymphoid organ, such as CCR7 was also positive in thymus, this naive B cell, a T cell. Then second one is a CCR5 and CCL13. CCR5 is present on the T cell and CXCL13 present on the B cell. They are important for the germinal center. So just remember for germinal center, you have to remember one receptor that is CXCR5 and one ligand that is CXCL13. Then for the TH17, ligand is CCL20 and CCR6 is a receptor. You just remember this one. So for the tissue, this cell has to roll, bind, and comes inside the tissue where they have to create the inflammation. That is called cell trafficking. Then tissue resident Suppose a patient is developing recurrent inflammation in the same organ. That means something there in the same organ. Or patient is doing good with the drugs, then suddenly relapse is there. Then this means there should be a memory. So T cells can become memory like also. So naive T cell, they got education in the lymph node also, secondary education, then becomes the effector T cell. Effector T cell goes through the diapedesis in the tissue, remain in the tissue, and in the tissue they becomes the memory. Example is RA, such as in blood, naive. Then in lymph node, they got education. Then they goes to the synovium, remain there, and helps in the formation of panas. Stay on how this memory B cell or tissue, so how this T cell becomes the resident memory B cell, resident T cell, because there are some growth factors like TGF beta, IL-15, IL-7, and important thing is CD69. CD69 is a marker for because it's an inhibitor of sphingosine 1 phosphate. So it inhibits the chemotaxis from that particular organ. So tissue resident memory B cell CD69 positivity. Sometimes CD123 positivity can be there. So the resident B cell in the synovium leads to the pannus. Local inflammation in the any resident B cells. So this is called tissue resident B cell that is important for the any rheumatological diseases. Then memory, same thing. T cell can become the T cell for the persistent because T cell antigen accumulation, then effector clonal expansion, that can lead to the memory. So if any their infection, second infection is there, then it will generate more rapid response. That is the basis of the vaccination also. Then T cell dysfunction. Suppose there is a constant inflammation. So T cell, after sometimes they lost their ability for the proliferation, lost their ability for the signaling, lost their ability for the cytokine secretion, and they start secreting some inhibitory molecules that is called T cell dysfunction or exhaustion. But this doesn't mean these are dysfunctional cell. These are non-functioning cell. They can functional in a different way. Energy means cell will be there but without any work. Exhaustion means this can convert into the again this can convert into the active cell. Senescence means old cell, and then finally convert into T regulator cell. So this is a pathway. Effector cell is there. Then early dysfunction can occur. Then intermediate dysfunction can occur. Then high dysfunction can occur. Then terminally dysfunction can occur. T regulatory T cell is a regulation function also. Suppose FOXP3 is there, then this T cell is important for the peripheral tolerance. So this is important for the peripheral tolerance also. So this is more FOXP3 CD25 rich. It can secrete the CTLA4. So suppose there is a T regulatory cell. This regulates the excessive immune system. But that doesn't mean if you have a excess T regulatory cell, that doesn't mean there will be no inflammation because T regulatory cells need to be active. So suppose this T regulator cells become dysfunctional, then there is there despite of T cell number like here, T regulatory function is not there. That is not in blood. Then repertoires. This is a T cell repertoires means different for the antigen specific T cells get change their receptor till they becomes the perfect for that particular antigen. That antigen is a selected antigen that is called and then becomes the clonality that is called the T cell repertoire clonality. Then how this T cell becomes the autoreactive. Suppose central tolerance loss, then T cell becomes autoreactive. Suppose peripheral T cell loss, then it can becomes autoreactive. So from the loss of tolerance, your T cell becomes autoreactive, and because of the genetics, epigenetics phenomena, environmental phenomena, T cell can becomes autoreactive. This is the fascinating part. T cell immune network. T cell doesn't work alone. T cell can work with B cell. Mostly T follicular helper cells work with the B cell. There is a CD40 ligand is important and IL-21. T and B cell can form the germinal center and they can create the inflamed organ. T cell can react to the myeloid cells such as the macrophage, and macrophage can present antigen to the T cells. So they can crosstalk. T cell can also crosstalk to the stromal cell, fibroblast cells. So T cells communicate with all other types of a cell, and T cell is regulated by the T regulator cells. So this is the T cell crosstalk. So we and now come to the CD8 cell. What I want to say, tell CD8 cell is not only a killer cell. CD8 cell can be inflammatory type also. So this one is a established phenotype. One minute. Sorry, T cell can be established phenotype like classical cytotoxic cell, memory cell, and regulated. But important new thing is this is called inflammatory type of T cell. They are Grange and K positive and dysfunctional T cell. This dysfunctional T cell found in lupus nephritis, and this Grange inflammatory T cells has been found in the synovium. So take home message is that CD8 cell is not always a killer cell. That they can behave like an inflammatory cell also. They can behave like a stem cell likes also. So this is their. So first, how they kill. How they recognizes the antigen through MHC1. Then how they kill? They kill through the perforins. Perforins create the pores in the pathogen, or Fas ligand that creates the apoptosis. Then gray enzymes. There is two types of gray enzyme. Gray enzyme B is responsible for the killing. Gray enzyme K is more responsible for the inflammatory action. So inflammatory action means IL-1, sorry, TNF alpha, interferon gamma, IL-17. So T cell can be memory T cell, can be regulatory T cell, can be inflammatory T cell, can have a direct contact, means physical contact action, or can work through the paracrine action such as interferon gamma, TNF alpha. So point is this: T cell is not only killer cell. It can behave like a regulatory cell. It can behave like an inflammatory cell. It can behave like a stem cell like. Or T cell can work through the direct contact. T cell can work through the paracrine fashion. So phenotype is not fixed. It's a dynamic, basically it's a dynamic action of a T cell. Then unconventional T cell. In the During the development of the alpha beta T cell in the thymus, 5% of the T cells in the early development they become the gamma delta. They comes outside the thymus. This is called gamma delta T cell. Then another type of the T cell is a MET, mucosal associated invariant T cell. Another is a innate induced natural killer type of T cell. All these variant of a T cell are very important in the spondyloarthritis group because they can secrete the IL-17 that can lead to the damage or inflammatory bowel disease, psoriasis, and the other diseases. This is important. T cell and B cell, I told you as already, such as this is a myeloid cell. Myeloid cell can be macrophage, can be some types of dendritic cells, neutrophils and neutrophils. Other. So, dendritic cell or antigen presenting cell can present antigen to the T cell and can activate the T cell, but the T cell can activate the antigen presenting cell. So this is called crosstalk. So they can potentiate each other, such as TH1. TH1's TH1 secretes the interferon gamma, which can stimulate the macrophage. Macrophage further can present antigen to the T cell, and through IL-12 they can form TH1. Through TH17 they can call the neutrophils. TH2 eosinophils. So means for the myeloid connection, it's not absolute, but remember TH1 macrophage, TH2 neutrophils, TH17 neutrophils, TH2 eosinophils. This is most important part in rheumatoid arthritis, T cell and fibroblast crosstalk. So once T cell got activated, they secreted different cytokines. So how many types of the T cell is there in RA, such as TH1, TH17, TFH? All these things they crosstalk with the fibroblast, THY positive, THY negative. THY positive is the inflammatory fibroblast. THY negative is a destructive, that is for the erosions. So T cell they secretes these cytokines TNF alpha, IL-1 gamma, interferon gamma, IL-17, GM, CSF, TGF beta. They stimulate the fibroblast. And then fibroblast continue or amplify the inflammation. So this is called fibroblast crosstalk. So T cell can talk to the B cell, T cell can talk to the myeloid cell, T cell can talk to the fibroblast. T cell can goes inside the tissue. T cell can reside, can remain inside the tissue, and that can be the reason for the long lasting autoimmunity. This is a vascular. Same thing, this T cell, they from the vascular, they can interact to the endothelium also. So this is the fifth part. So T cell can interact to the endothelium, can interact to the B cell, can interact to the myeloid cell, can interact to the fibroblast, and all these tissue microenvironment is important. So this is a tissue microenvironment as I told you. So T cell can interact with all these structure and they form the tertiary lymphoid organ in the tissue and that tissue becomes the inflammatory organ. So this is for the rheumatoid arthritis. So let's comes to the last two diseases. How many types of T cell we read in rheumatoid arthritis? Th1 is talking to the macrophage, that leads to the granuloma and pannus formation. That is Th1. Th17 can call the neutrophils, can lead to the acute inflammation. Th17.1 is a Th17 cell which has a capacity to secrete the interferon gamma also. So this is also there. T follicular helper cell talk with the B cell and can create the antibody. T peripheral helper cell is a type of T follicular helper cell without CCR7 positivity, then can talk to the B cell. T regulatory cell, memory cell, tissue resident B cell. So we have Th1, Th17, Th17.1, TFH, T peripheral helper, T regulator, T memory, T tissue resident. This is for the CD4 subtype. And CD8 subtype, we have the classical CD8 that is killer CD granzyme B positive, granzyme K positive is also there, that is for the inflammatory. Then tissue resident memory B cell can also be there. Then second thing, TH17 is involved in RA, but not only TH17. So these are the these are the T cells, and TH17.1 is more important in RA. Then this is the spondyloarthritis group in which different types of the T cells like TH17, T killer 17, gamma delta T cell, MET cell, tissue resident memory T cells, they secretes the IL-17 and TNF alpha, and that causes the spondyloarthritis. So thank you. I just want to cover this uh, T cell in half hour, but okay. Thank you so much.
Okay. Thank you, doctor, for sharing your expertise with us and really making the session so insightful and such complex, topics of T cell thymic selection and T cell education. What you gave to us, such complex concepts, you've tried to narrow down into very simpler form. So once again, thank you from all of us and for this great learning. Indeed, it was a great learning for all the participants. So let's just go through the question answers session if there are any available. Doctor, we could find one or two questions are there. It would be great help for us if you could answer. Um, like Doctor, there was a slide 4 in which I think you showed some three signals of the T cell activation. Right. So there's a question regarding that. That if all the three signals are present, what ultimately decides or determines whether T cell becomes an effector cell or a memory cell or a regulatory cell? So what is your
These three signal means first T cell has to be recognized by the some antigen. The first signal is recognition. Second is for the permission. Permission is for means T cell got energy, some activation. Then third part is a polarization. Polarization, as we know, T cell is not only CD4 or CD8. There are a variety of a T cell. So on the basis of the cytokines, T cell can convert into the T helper 1, T helper 17. So why this is important? Suppose there is a one disease in which, if we go for the old hypothesis, which is moving towards the more TH1, then it will go towards more RA type of picture. if T helper cell goes towards the TH17 type of variety, then it will go towards a spondyloarthritis group more. It's not like a clear cut, but broadly. If suppose T helper cell convert towards the TH2, then it will go towards the asthma or eosinophilic granulomatosis, means some allergy part. Similar way, suppose there is sustained inflammation, then our body needs to stop also. So this cytokine becomes in that situation, cytokines become in that concentration. So T cell becomes the regulator which start inhibiting Th1, Th2, Th17. So this is a combination or cocktail of a cytokine which decide in which direction T cell will move.
Fine, doctor. Thank you so much. Doctor, this is a concept of fibroblast heterogeneity. Can you just
Actually, this is very fascinating. From 2024 till on, initially what we thought, rheumatoid arthritis is a problem of synovium. Okay, in synovium we have synovial cell. Synovial cell has two variety, macrophage and fibroblast. If you ask even anybody, go back and remember from your 12th or first year something, fibroblast is a structural cell. That is our concept. Fibroblast is doing what is some structural part, and macrophage is an inflammatory cell. So if we want to choose among the macrophage and fibroblast, which is dominating cell in rheumatoid arthritis, synovium, then automatically we will think it's macrophage only, because macrophage will do all this destruction. But in reality, it's different, very different. This fibroblast, this macrophage, T from the T cell, macrophage and B cell, somehow this fibroblast from this resting or repairing properties converted into the inflammatory variety. Means first property was this is repair. Second, then they becomes the inflammatory. And third was the destruction. So fibroblast has a different phenotype. It will depend upon the context. Suppose healthy person is there, function of a fibroblast is repair. Suppose autoimmunity is there, then it can create severe pain or inflammation, or it can create the damage, destruction. So this is called this fibroblast heterogeneity. Means you can't pinpoint fibroblast means only this one. No, it can change from this to this to this.
fine, doctor? Thank you so much. And if any other questions are there, you are requested to drop in the chat box, and definitely the doctor will assist for the same. So, with this, doctor, with your permission, we can just come on to the closing of the session. And thank you all the participants. And today was a seventh series, second session of the series that we are taking forward. And soon we'll be coming with the eighth part, and all the videos and everything starting from 1st to 7th have been uploaded on the YouTube page. So if any clarity required, you can all anytime you can just visit the sessions of the doctor, and doctor is always ready to assist for the same and to take this things forward. So I think, doctor, with your permission, we can put a close the session for today.
Thank you. Thank you. Sure. Thank you.
Thank you so much, all the participants. Thank you, doctor, and we'll be soon coming up with the next session. Until